My name is Rachel Silvio, and I'm going to talk to you today about erythrocytes, blood types, and transfusion reactions for the cardiac II and blood presentation. Let's start out our lesson today with an interesting fact. There are about 5 billion red blood cells per 1 milliliter of blood. Since the average adult has 4.5 to 5.5 liters of blood in their body, that equates to 22.5 to 27.5 trillion red blood cells. Wow. Erythrocytes are also called red blood cells or RBCs. They are the perfect example of how structure is vital for the function. There are 3 structural components of red blood cells that allow them to carry out their most important function of carrying oxygen. The first is their shape. These cells are flat, disc-shaped cells with indentions in the middle on both sides. The shape allows for a larger surface area for the diffusion of oxygen from blood plasma into the cell through the cell membrane. Because the cells are also thin, diffusion is quick. The second structural component is their flexible membranes, allowing them to squeeze, even single file sometimes, through tiny capillaries. The diameter of a red blood cell is normally 8 micrometers, yet the cell can fit through 3 micrometer capillaries without rupturing. This adaptation is crucial for the blood cells to be able to carry oxygen to all tissues of the body. The third unique characteristic is that they are composed of hemoglobin. Because red blood cells do not contain a nucleus or other organelles, they have room for about 250 million hemoglobin molecules. The structure of hemoglobin is also very important to its function. It is comprised of 2 parts, globin and heme groups. The globin portion is a protein made up of 4 folded polypeptide chains. Each one of these polypeptide chains binds to the heme group. Heme groups are iron-containing non-protein groups. Each of the 4 heme groups can bind to the 1 molecule of O2, allowing for up to 4 O2 molecules for each hemoglobin molecule. Oxygen is poorly soluble in the plasma, so 98.5% of oxygen in the blood is bound to hemoglobin. Oxygenated blood is reddish because of the iron and oxygen. Hemoglobin can also combine with 4 other compounds. The first one is carbon dioxide, CO2, carrying it from the tissues in the body to the lungs so that the individual can exhale it into the atmosphere. The second compound is the acidic hydrogen ion portion of ionized carbonic acid, which is generated from CO2 in the tissues. Hemoglobin buffers this acid to minimally alter the pH of blood. The third compound that can be carried by hemoglobin is carbon monoxide, CO. If this gas is inhaled, hemoglobin will bind to CO instead of oxygen, causing CO poisoning. Because carbon monoxide binds to the hemoglobin in a straight line, carbon monoxide is a better binder and becomes a competitive inhibitor to oxygen. The oxygen, as seen on the left, binds at a slight angle, since one oxygen atom binds to the iron and the other binds to the side chain of a histidine amino acid. The last compound that can bind to hemoglobin is nitric oxide, NO, which is a vasodilator. Released in tissues, relaxes or dilates arterioles, stabilizes blood pressure, and ensures delivery of oxygen. Just recently in 2020, hemoglobin research was conducted on the effect of carbon monoxide exposure on hemoglobin mass. Their hypothesis was if oxygen binding sites on the hemoglobin molecule are blocked by administering low-dose carbon monoxide, then hemoglobin mass, HB mass, and VO2 max will increase similar to the effects of altitude. The control group was 11 healthy and moderately trained males who inhaled a placebo. The experimental group was 11 healthy and moderately trained males who inhaled a CO2 bolus 5 times per day to increase the HbCO concentration by roughly 5%. The results were the HB mass increased in the experimental group by 4.68% in week 3 with a p-value less than 0.001, which is statistically significant. For the following 3 weeks, the level maintained. VO2 max increased by 2.84% immediately after being exposed to the CO with a p-value of less than 0.1. The study showed a positive relationship of VO2 max to the change in HB mass with a p-value of less than 0.001, which is extremely statistically significant. This positive correlation is shown in the graph on the slide. The conclusion of the study was that it showed that continuous exposure to low-dose CO increased the HB mass and VO2 max by erythropoietic processes, which is the production of red blood cells from erythropoietic stem cells involving differentiation and gene expression. Erythrocytes have two different enzymes that assist in their function, glycolytic enzymes and carbonic anhydrase. Glycolytic enzymes generate energy needed for active transport mechanisms for ionic homeostasis or balance. Since red blood cells do not have organelles such as mitochondria, they must solely rely on glycolysis for ATP production. The second enzyme is carbonic anhydrase, which assists in carbon dioxide transport. Catalysts in reaction that converts CO2 into HCO3 bicarbonate ion so that it can be transported. An antigen is a large molecule, usually a protein, that triggers a specific immune response against itself when it enters the body. One example would be if a bacterial cell enters into another organism's body. It is a foreign object. On the surface of the bacterial cell would be antigens of a specific shape. The body responds against this bacteria to try and fight against it. Some white blood cells will recognize the antigens and make antibodies against them. An antibody is a protein that binds to the antigen based on a specific shape and leads to destroying that antigen. There are 4 blood types in the ABO blood groups: type A, type B, type AB, and type O. The surface of the red blood cell has antigens which are inherited. A blood has A antigens, B blood has B antigens, AB blood has both antigens, and O blood has neither antigens on the surface of the red blood cell. A blood has anti-B antibodies, B blood has anti-A antibodies, AB blood has neither of these antibodies, and O blood has both anti-A and anti-B antibodies. Type O is the universal blood donor because O blood does not have A or B antigens, so no other blood type will attack this blood. Type AB is a universal recipient because type AB blood does not have anti-A or anti-B antibodies, so no other blood type will attack. Rhesus Rh factor is an inherited protein on the surface of the red blood cell. Rh-positive blood is blood with the Rh factor or antigen. Someone with positive blood can receive negative or positive Rh blood through a transfusion. Rh-negative blood has no antigen. Someone with negative blood can only receive Rh-negative blood through a transfusion. Antibodies are not naturally occurring against Rh factor. Anti-Rh antibodies are only produced if Rh-negative blood is exposed outside Rh-positive blood, such as during birth. Rh incompatibility is when a pregnant mother is Rh-negative and the child she's carrying is Rh-positive. When the woman is exposed to Rh-positive blood from the fetus, her body creates anti-Rh antibodies, which could possibly attack the blood cells of the fetus, leading to serious health risks such as anemia, jaundice, or possibly death, often in a future pregnancy. A medication can be given to an Rh-negative woman during pregnancy to prevent her body from developing anti-Rh antibodies. There are about 23 other minor human erythrocyte antigen systems, comprised of hundreds of subtypes. In a non-emergency setting, the medical team can check transfusion donor by mixing blood and ensuring there is no clumping. If a person is given blood of an incompatible type during a transfusion, their body could negatively react. One reaction is when the donor's antibodies fight against the recipient's antigens. This particular reaction is mild, as the donor's antibodies are diluted compared to the recipient's. The second reaction is when the antibodies in the recipient's blood reacts to antigens on the donor's erythrocytes. This reaction could be agglutination or hemolysis. Agglutination is the clumping which can clog small blood vessels. Hemolysis is when the red blood cells rupture, releasing large amounts of hemoglobin and causing kidney failure. Both agglutination and hemolysis can be fatal. The picture on the right of the screen shows what happens when different blood types are tested for antibodies. For example, O positive has both anti-A and anti-B antibodies, but not the anti-Rh antibodies, so the results would be no clumping for the anti-A and anti-B antibodies, but clumping for the anti-D, which tests for anti-Rh antibodies. Let's do another one. A negative blood has anti-B antibodies and anti-Rh antibodies since it is negative. The results will show clumping in anti-A, no clumping for anti-B and anti-D. Now that we know how to read the results, let's do an unknown. The picture on the slide shows the result blood typing for 4 patients. Let's figure out what blood type patient C is. There is clumping for anti-A antibodies and clumping for anti-B and Rh factor. So this patient has anti-A antibodies, no anti-B antibodies, and no Rh antibodies. So they have B positive blood. Let's go back to our blood typing picture and compare the results for patient C to blood type B positive. See, no clumping for anti-A antibodies and clumping for anti-B and anti-Rh antibodies.